I have so many windows open.
Yep.
Okay, people are starting to file in. All right, it is 7:02 EST, and so I'll start by introducing myself. I'm Candace. I am one of the organizers for the BioCore Network Summer Conference, and I also go by Kraken on the Discord chat. And so I want to introduce to you Dr. Renu Verma. She is an excellent molecular microbiologist. She has more than 11 years of experience in infectious disease. She does a lot of work with novel strategies and developing low-cost diagnostics. And so she's here to talk to us about, about the COVID-19 pandemic and the rise of global collaboration and the lessons learned through this. So welcome.
Thank you so much, Candice. It's a pleasure to be here. I'm just gonna quickly share my screen. All right. Yeah, so I'll be talking about some of the work we have been doing here on COVID-19 at Stanford. But before we move forward, I would just like to talk about where we began and where we are after 1 year of pandemic. Am I audible enough?
Yes, you are very audible.
Okay. So, yeah, March 11, 2020, WHO declared COVID-19 as a pandemic with more than 100,000 cases and 4,000 deaths in 114 countries. And by mid of March, we already had a variant circulating. And by August, Sputnik 4 COVID-19 vaccine receives approval by Russia. And by September, global death toll reaches 1 million. And on October 22, FDA approves remdesivir for the treatment of hospitalized cases of COVID-19. And by December 2, UK becomes the first country to approve Pfizer COVID-19 vaccine. By January 15th, global death toll reaches 2 million. And after 1 year of pandemic, we did a lot, but then the pandemic continues with 118 million confirmed cases, 2.6 million deaths, 66.7 million recovered, and 70.5 million fully vaccinated, and still counting. So apart from all the challenges which COVID-19 pandemic has given, it has changed the way scientists or researcher work globally, and there have been more and more international collaborations. An earlier survey before pandemic by the US Office of Science and Technology showed that about 1/3 of those who collaborate internationally, they are originally from another country and were connecting with colleagues there. Another third were collaborating with someone who had worked in one capacity or another with the United States. So basically, scientists were maintaining connections that had been fostered locally, a trend that continues even today. And as many as 90% of these international collaborations began somehow face-to-face or side by side. And COVID-19 pandemic changed this trend for good. A 3% increase in collaborations between individuals that had never worked together was observed. And these collaborations involved scientists from different disciplines that showed signs of greater novelty. If we look at overall picture of the rise in collaboration, we observed an increase in collaboration involving more than 3 or more nations, which accounted for 30% international collaborations and 7% of all the articles which were cited. It was observed that when 2 or more nations are collaborating together, that research is cited more. So when the pandemic began, like all other research institutes, there were a rapid response from the Stanford research community as well. As many as 68 new projects were started at different divisions, which included from testing to develop new diagnostics, to study the disease transmission, to work on vaccine and treatment, to study the epidemiology of the disease, to work on data science and modeling of transmission, work on immunology, and so on. At Andrew's lab, we also collaborated and work on different projects. I'll be just focusing on developing some novel assays to study the infectiousness through exhaled breath. So from the beginning of pandemic, infection through aerosol transmission has been in debate until recently, when there were some reports. So earlier reports or guidelines by CDC told that defined a close contact as an individual who spent at least 15 minutes within 2 meters of an individual with COVID-19. So these guidelines were followed by different institutes and schools, and a school in Iowa, teachers were told to get students up and walking around every 12 to 14 minutes to avoid exposure. And the Iowa Department of Public Health stated that only those exposed to the virus for 15 consecutive minutes within 6 feet need quarantine or exposure. So these were the earlier guidelines. However, there was no empirical evidence of this 15 minutes rule, but this was widely followed until there was infection reported among several NFL players, and it was after observing that CD and when they investigated the cause of infection, that they found that none of the players were actually exposed for 15 consecutive minutes. And video CCTV showed that over a 24-hour period, they were at least exposed for 15 minutes. So based on these observations, CDC changed guidelines and told a close contact is an individual who spent at least 15 minutes over a 24 hours period within 2 meters of a COVID-19 infection. So these are the current guidelines of CDC for exposure, where if you have been in contact with a COVID-19 infected person for 15 minutes within 24 hours, you may be at risk. Even now, there are no empirical evidence and to decide that cutoff, and there are several research gaps which we have been observing from the beginning, and there are several questions which need to be answered to redefine these guidelines. So the major research gaps were 15 minutes rule has been widely used to inform contact investigation in community as well as in healthcare settings based on a premise of average risk, though the empirical basis of this specific window is limited. There are no direct measurements of variation in exhaled breath SARS-CoV-2 abundance. So we still didn't know for 15 minutes how much amount of virus is released in exhaled breath. And this was mainly due to lack of a flexible or sensible tool which can capture SARS-CoV-2 from exhaled breath to define risk or quantitatively assess it to tell the determinants of variation. We hypothesize that just based on these 15 minutes window, it may not be enough to define the risk, and there could be other factors such as inter-individual variabilities, age, or a person's lung health, and whether a person was talking or not talking during exposure. Keeping these research gaps in mind, we designed a novel mask tool, which was easy to use. It was cost effective. It had minimal discomfort, and no technical expertise was required to collect the sample. There was we didn't need any liquid sample collection. And all we did was, there is a special membrane which is called as gelatin membrane filter, which is derived from LML protein. We fitted this gentle membrane filter on a disc, and then we connected it to the mask. You can see the prototype here. And this mask directly collected the exhaled breath sample on the disc. And these samples were transported to the lab at room temperature. And all we had to do is just add a viral stabilizing media. Another important property of this membrane filter is it maintains the viral viability. So that gives us opportunity to study the infectiousness or intactness of the viral particle. So after designing these masks, we validated our mask recovery of viral RNA based on the nasal swab, which is currently used as a gold standard. We collected swabs, these spike swab with the same amount of virus, at different copies of virus with the serial related samples, and spike same amount of virus on the mask, and observed that the recovery of the viral particle during extraction from mask was equivalent to what we were getting from swab, and the limit of detection for both the assays was as low as 10 copies per mask or per swab sample. After validating our mask design, we further moved to validate our hypothesis. For that, we recruited 97 COVID-19 positive individuals, and these individuals from Stanford Hospital, those were admitted but didn't require oxygen. And there were individuals from outpatient clinical trials between September 2020 to March 2021. We divided these individuals into two groups. Group A wore a mask for 30 minutes, and they were not given any instructions, so they were allowed to talk or not talk without any further prompting. On the other hand, group B was instructed to wear 2 masks. For one mask, they were instructed to talk with the interviewer or a family member for 15 minutes. And for another mask, they were instructed not to talk. The idea was to see that what would be the viral load when a person is wearing mask for double the time while not talking, or just not talking or talking. So after we had these masks collected, we extracted the RNA, and for our further analysis, this was the overall study population characteristics. So we had patients from different age groups, from 40 to 60. In group B, we had even individuals with the age group in 30s. And we had few patients didn't have any symptoms at all. And most of the patients had symptoms for 3 to 6 days. And these were the symptoms, common symptoms, which were observed in symptomatic patients which were recruited for the study. So these are some key findings. Our mask showed a very high sensitivity to capture exhaled breath, virus from exhaled breath. Previous studies which use a different sample processing and capture method have reported a maximum 38 to 40% sensitivity, which is why these assays cannot be reliably used to assess the quantity of viral particle in exhaled breath. On the other hand, our assay showed a quite high sensitivity, and we observed that viral copies which were observed in breath moderately correlated with nasal swabs. So when we collected the viral samples on exhaled breath, we also took nasal swab from same patients just to see what is the correlation. If a patient has higher viral load in nasal swab, does that mean he would be shedding more virus in exhaled breath, or there can be some external factors as well? We observed that there was a moderate correlation between nasal swab and viral copies. So, and we concluded that there have to be some additional factors which are adding on the amount of viral that are released in exhaled breath. Coming to the talking and no talking groups from same individual, we found that the positivity rate was significantly higher when an individual is talking. So for the same individual and same amount of time, the positivity was 59% when a person was talking, as opposed to 42% when he was not talking. Even the viral copies, number of viral copies which were released were quite high. We developed our assay and developed our own standard curves using plasmid. Plasmid I'll show you in later slides to quantify exact quantification of viral copies. And we found that the total viral copies which were observed in talking were substantially higher than the no talking. In fact, for few samples, we observed as high as 5 million copies of viral particles released while talking. And we further investigated whether viral shedding in exhaled breath is related to the number of human cells which are released in exhaled breath. We used human RNase P gene as a marker, which is a housekeeping gene, and we found that the number of human cells which are released in exhaled breath are following the same trend as the viral particle. And in case of 15-minute talking, it is releasing the maximum amount of virus. As you can see, when a person is not talking, when a person is talking for 15 minutes, that would be half the amount of time, 30 minutes, he's still releasing more viral copies. So based on these, the guidelines where CDC says that a 15 minutes exposure is enough, it would need some other criteria as well, which could be whether that person was talking or he was singing or how close he was. So this was the first quantitative experimental evidence of how many viral copies are actually released when a person is breathing or talking. And none of these patients were coughing. So we were recording that data as well. There was a study which showed that the amount of cells or bioaerosol released also depends on the lung health and age of an individual. So we compared that with the viral load and RNase-P abundance, and we did found a correlation that people with higher age were releasing higher viral particle with 15 minutes talking than the younger individuals. So after confirming that, we further explored the utility of our mass sampling to study different variants in SARS-CoV-2. And for the first time, we demonstrated that exhaled breath sampling can also be used for the variant surveillance. We randomly picked samples from no talking group, and 30 minutes. And then we compared the quality of whole genome sequencing data with the nasal swab. And we found that majority of our samples, they passed the QC, and majority of 71% of samples had coverage depth as high as 100x. And for all the samples, we could reliably detect variants of concern. So this is another important application of the tool that we have developed here, which can be used for other infectious diseases as well. And we found that median coverage depth was correlated with the viral load. So in conclusion, the assay the tool that we developed, it had higher sensitivity than previously reported studies. And we found that individuals shed several-fold higher viral particles while talking. And this is first quantitative evidence that on the amount of viral particles which are shed in the exhaled breath. Our mass sampling tool can further be used as for variant surveillance, looking at the whole genome sequencing data. And another major application for this tool can be exhaled breath pooling, where these filters or masks can be installed in front of offices or closed spaces. And individuals can be allowed to talk for a minute, and then at the end of the day, you just collect that sample and pool it, and then use it for the surveillance. And this tool, of course, can be used to study other respiratory pathogens like influenza or TB. So this prototype is a proof of concept that this tool can further be used for other applications as well. But overall, there is more studies are required to further investigate the risk and the amount of viral load which is shed. So I'll quickly move to another important and very interesting study we started early during the pandemic, which was development and validation of molecular viability assays for SARS-CoV-2. So one of the major tests which were used to detect virus was real-time PCR, RT-PCR assay, which detects the viral RNA. And several studies and clinicians were reporting that even after recovering from the disease, patients were tested positive for real-time PCR for several weeks, because of which they were told to quarantine for longer periods. However, there were possibilities that real-time PCR, because it's a very sensitive technique and it can even amplify a single viral copy, there was a possibility that the sample was detecting some residual viral particle which was not part of an active infection. So there was a need to identify a novel strategy or some markers to detect the actively replicating virus. And because the other way to tell the infectiousness is to culture the virus, which requires PSL3, and it is quite resource intensive. So that was the need of the hour, and mRNAs or pre-ribosomal RNAs have been used for to detect the replicating bacteria or viruses for in other diseases. And so there was a growing interest on using subgenomic RNA to detect replicating SARS-CoV-2. And in fact, in early 2020, there were some studies where sgRNA was detected and it was correlated with culturable virus, and clinicians were using sgRNA to decide if a person is infectious or not. Um, to talk before sharing the data, I'll just want to give you a brief introduction of what sgRNA is. SARS-CoV-2 follows a unique replication pattern where once it enters the host cell, it produces several small SG or subgenomic RNAs, which are then translated into functional or structural proteins. These subgenomic RNAs are not part of the final viral packaging. And so therefore, it is presumed that if we detect the subgenomic RNA, we are essentially detecting replicating virus. And also genomic RNA is actually protected by viral capsid, which is why it has a longer half-life, and due to which it was detected for a longer period in clinical samples. So even when the virus is not replicating, the dead virus was protected by the viral capsid. However, if sgRNA are not part of the final viral packaging, they are exposed to the lysis, and they shouldn't be. So the half-life of sgRNA shouldn't be longer. And once the virus is dead, we should not detect sgRNA for longer periods. Keeping that hypothesis in mind, we designed some subgenomic RNA assays. So another interesting fact here is SARS-CoV-2 genome has a unique leader sequence at the 5 terminal end of its genome. So it's called as jumping RNA gene synthesis, where leader sequence fuses with all the genes, and then you have these smaller subgenomic RNA. So you can see you have N gene, 7A, 7, 8A, all these genes. The important thing to note here is that every subgenomic RNA has a leader sequence, which is common, which is fused. So if the concept of the entire test is that if we plan to design an assay which can amplify only genomic RNA, we can amplify any of these sequences for N gene or E gene. However, if we want to detect sgRNA of N gene, we have to amplify the leader sequence plus sequence of this gene. So based on that, we designed sgRNA assay and compare it with both N gene and sgRNA to see the efficiency of our assay. So our assays could detect as low as 1 copy per reaction of a viral particle. And so we use that assay for our initial validation to see what we are detecting is actually subgenomic RNA. So we use a couple of clinical samples from Lambda and Favi for our clinical trial. And we found that all the sgRNA that we detected through real-time PCR 100% matched with the leader sequence. So once that was done, we further try to investigate. So our hypothesis was if sgRNAs can be used as a viability marker, their decay kinetics should be different from the genomic RNAs, because genomic RNAs are protected by the viral capsid. So for that, we recruited 205 patients from 2 different clinical trials at Stanford, and we collected 536 longitudinal samples. From Lambda trial, we collected samples on day 1 when the patient walked in, and it will be expected to have highest viral load, and then day 4 and day 6. We measured genomic and subgenomic RNA at day 1, day 4, day 6, and even for Favi, we measured genomic and subgenomic RNA at all the time points. From the previous clinical studies, we already had information that genomic RNA is detected for longer periods, even for weeks. And we found that in both the clinical trials, genomic and subgenomic RNA had very high correlation. So with days of infection, we found that genomic and subgenomic RNA both are decaying at the same rate. And then another interesting observation was sgRNA was not detected for samples for which genomic RNA CT was high. So basically, when a patient had lower copies of genomic RNA, subgenomic RNA was not detected. And previous studies, the way they concluded that we are detecting as genomic RNA and not subgenomic RNA, and the patient is no longer infectious. We hypothesized that we are not detecting sgRNA because of the abundance and not because a patient doesn't have a replicating virus or vice versa. And then we compared our data, proportion of positive with the days of symptoms. And if subgenomic, if a patient is not harboring any replicated virus, we should see a decline in sgRNA with the symptom onset. Even by day 21, sgRNA levels were comparable to genomic RNA, further suggesting that both have similar half-lives. To study that, we investigated the decay kinetics of clinical samples, and we found that even in both the clinical trials, when patients were given different drugs, the decay kinetics for genomic and subgenomic RNA was similar. And that made us think that maybe subgenomic RNA is not a good marker, and it is giving a false negative result because of less abundance, because of less assay sensitivity, and something else is going on in terms of viral stability. Because if you look at the molecular mechanism, RNAs are highly unstable and dead cells are full of RNases, so it should quickly degrade. Something else is going on. And then a study was published where they sequenced subgenomic RNA, and there was a hypothesis that there is a possibility that subgenomic RNAs are protected with some membrane vesicles. So this study, they treated the cells with detergent to lyse these membrane vesicles, and then they observed that sgRNA was degrading. So even then, there was no experimental proof to show that sgRNAs are protected or not. To validate that, we infected human lung epithelial cells with subgenomic RNA. And once the cells were infected, we treated them with remdesivir or a control. So remdesivir is an RNA-dependent RNA polymerase inhibitor. So it stops the viral replication. So we hypothesized that once we treat the cells with a drug, viral replication will stop. And we started quantifying the genomic and subgenomic RNA from there. If subgenomic RNA is not protected, it should decline faster than genomic RNA. And we started quantifying genomic RNA at different time points up to 50 hours post-treatment. And we found that subgenomic and genomic RNA followed the exact same trend. And this was another evidence that subgenomic RNA are actually protected. So from this study, we concluded that for other pathogens, mRNA could be a very reliable viability marker, because the mRNAs are exposed and they are just the replicative intermediates. However, in case of SARS-CoV-2, the mRNAs are actually protected by a double membrane structure. So just like viral capsid, subgenomic RNAs are also protected and will be detected for longer periods than expected. So these assays would give a false negative results if used as a viability marker. So our study provided first clinical and experimental evidence on cell lines, proving that further markers need to be explored to study sgRNA. And we also just try to investigate the abundance of sgRNA, because we used a novel approach and a different assay where we compared N-gene sgRNA with N-gene genomic RNA. Earlier studies were comparing E-gene sgRNA with N-gene genomic RNA. So it was because there was a difference in abundance, they were detecting genomic RNA and now detecting subgenomic RNA, and concluding that patient is no longer infectious. And and then just to study the sample quality, we used human RNase P gene, and we found this was fine. And this was a very interesting fact, which was released on October 10, 2020 by the White House, physician to the president. When President Trump was infected with COVID-19, SARS-CoV-2, this letter was released, and they had performed subgenomic RNA tests to say that he is no longer infectious. And at the same time, there are several commercial kits available in the market which use subgenomic RNA as a molecular viability markers. And these kits have been sold, and based on these, decisions are made whether a person is infectious or not. Our findings for the first time actually conclude that more research and more investigations are required before we actually commercialize or release any kind of assay in the market, which may directly impact the clinical decisions. And we found that abundance of sgRNA and gRNA in clinical samples were highly linearly correlated, and decay of sgRNA and gRNA in clinical samples did not differ. And like I mentioned earlier, previous reports of lower positivity of sgRNA in clinical samples is likely due to lower abundance. And then, of course, remdesivir treated cells also showed the same decline rate. So overall, there is no evidence to support the transcriptional or replication intermediates as providing orthogonal information to genomic RNA abundance in inferring infectiousness or active infection. So this study was recently published in Open Forum Infectious Diseases. If you want to further have a look at the data, or you have any questions, you can reach out. And this is my last slide, and this is kind of disturbing. This has been this has been viral on Twitter for some time. We recently completed 100 years of BCG vaccine, and my major work is on tuberculosis, which remains the biggest killer of the century, or not even the century. You can just say biggest killer so far. And if you look at the trend here, it's quite disturbing. In 1882, TB bacteria was first discovered. COVID was discovered in 2019, end of 2019. In 1921, we had first vaccine. In 2020, we had first vaccine. In 100 years of, even after 100 years or more, there is only one vaccine which is effective in children. And so far, we have no effective vaccine for TB. And by June 2021, we have 18 vaccines for COVID-19. And number of vaccine candidates so far, after 100 years of research, is just 14 candidates, whereas we have 109 vaccine candidates for COVID-19. This is a very unfair number here. It's just 0.117 billion dollar investment on vaccine on TB, whereas we have 107 billion dollar investment on COVID-19 vaccine. Now, it's TB is a silent killer, which is, it's very unfortunate that drug-resistant bacteria is on the rise. So you see, in 2020, there were 1.9 million deaths due to TB, and 2 million deaths due to COVID-19. So this is the biggest lesson we have learned from this pandemic, is that the inequity in the fund distribution and the focus these infectious diseases they get. I mean, if the entire scientific community has enough funds and everybody comes together, I mean, the disease which has been lingering around for so long, it can be eliminated forever. And this holds true for other infectious diseases as well. And I really hope that the lessons we have learned from this pandemic will be applied on other infectious diseases as well. I would like to thank my mentor, Dr. Jason Andrews, my colleague and friend Eugene, Catherine, Nick, Bonnie, and all other members of from School of Medicine, and the entire team from Lambda, Favi, Regeneron, and Chemostat clinical trials. I would like to thank Bill Gates and Ming Foundation for the funding of this research work. And I'm open to questions now. Thank you.
Thank you so much for that really excellent talk. I do have some questions from the audience regarding, excuse me, regarding the first portion of your talk. Somebody asked, how difficult is it to run a study like that during a pandemic where you have people who are talking with their masks on? Like, what type of additional regulations do you have to put in place when there is a pandemic going on?
Oh, that's an excellent question. Stanford had been very strict when it came to this. Of course, proper PPE and training is first thing. All the individuals which were involved in clinical research, they had to undergo training and then some exams. And after training, they were under observation, and then we had to weekly get tested. And then we had every day before we check in, we had to report our daily health and symptom status. And at the clinical trial, the entire sample collection and everything was taken care and overlooked by physicians and professors and the clinical trial PIs who were highly trained. So yes, yeah.
Okay. And another person has asked, how often are the chances that some other infection has been mistaken for COVID-19?
That's a good question. So the chances are higher because COVID-19 symptoms are very similar to some other diseases as well, some flu symptoms and all that. Luckily, we have some good preliminary tests for which we get results very quickly. So usually the usual protocol would be to have a questionnaire where they ask how patient is feeling and then how symptoms are matching. And then based on that, doctors or physicians usually order a couple of other tests as well if they are suspecting any other respiratory or infectious disease.
And so in the process of developing these diagnostics for viral infectious diseases, how different is it from developing diagnostics for bacterial infectious diseases?
Excellent question. It's slightly different. One reason is it's easier to culture bacteria because it doesn't need any cell line. You don't need a host cell line. So to establish a cell line which can infect the virus itself is a huge challenge. So coming to developing the diagnostics, most of the viruses they are RNA viruses, and then so they follow a slightly different strategy than bacteria, which have a bacterial genome which essentially has DNA. So in terms of designing the experiments from the beginning, where we need standards or some cell line experiments, I think having diagnostic for the virus study is still a little bit challenging. than bacterial studies. And yeah, I think for the diagnostics, getting procuring standards would be another issues, because once you before you develop a diagnostics, you have to actually assess it against different panels, and you need to have synthetic DNA or RNA. So I recall that we were really struggling to get the viral standards to assess our assays, how many viral copies we are detecting for SARS-CoV-2. Whereas it's slightly easier for the bacterial genome, because you have DNA, which can be easily synthesized, and then you can just amplify it. I hope I answered your question.
Oh yeah, absolutely. And sort of related to that, for a bacterial culture, how do you validate whether it behaves the same way in the dish as it would be wild?
Okay. So for that, you need to. So for having host pathogen interaction, that's an entirely different study. So when you are developing a diagnostic, you just want to you are trying to investigate biomarkers. So it's mostly biomarker discovery, where you can have the DNA or nucleic acid of the bacteria. But when you're talking about host pathogen interaction, you need to have cell lines which are adapted for that virus or bacteria. In case of TB, we have THP well cell lines, which are for macrophages. So based on those cell lines, those are closer. And of course, we have animal experiments. We have mice studies, and then macaques. So based on what your interest is, what you are looking for, are you trying to study host pathogen interaction or pathogen evolution within the host? I think possibilities are multiple. So yeah, if you could give me a particular question, I could just answer in that direction. Yeah.
Okay. And then another question I have from the audience is, even with a high false negative rate for the sgRNA, does it compare with the current methods of using, well, I don't know what the current method is, using qPCR, and can using the sgRNA be used clinically?
Oh, that's a very clever question, actually. And I think that person is trying to ask is that instead of using other genomic can we use subgenomic RNA as a marker, just like genomic RNA? The answer is we validated that the sensitivity is slightly lower, because the abundance of sgRNA is slightly lower than genomic. See, there is one genomic RNA and multiple subgenomic RNA, and all will be amplified when we use a genomic RNA panel, whereas subgenomic RNA is subset of genomic RNA. So the overall abundance would be lower. So if we want to use subgenomic RNA as a marker for your diagnostic assay, of course you will detect the virus, but then you'll be compromising the sensitivity.
Okay. Well, that's I like that. And then I have one more question. So this is related to your other work. So how common is it for research labs to form collaborations with diagnostic companies like Cepheid?
Yeah.
Cepheid. And how is that collaboration approach? Like, who approaches who?
All right, that's at Stanford. I mean, it's a part of the culture, having startups and collaborating with different companies. My research is mainly focused on translational research and developing assays. So it was very common. I mean, when we initiated this idea of developing a pharmacogenomic assay, we approached Cepheid. But we do have other labs who are approaching different companies or having their own startups to translate whatever they are doing. So it's to answer your question, it's pretty common.
Okay. And then another question, how can we build a momentum to get better funding and support for finding a cure for other infectious diseases? And how can we avoid waiting for the next pandemic?
Answering your first question, I don't know. That's so tricky. I think just having that awareness would be a start. Like I have been working on TB for so long, and it really scares me. But TB is more prevalent in developing countries, and surprisingly, when I interacted with a couple of people here, not from Stanford, just outside, there was this misconception that TB is gone. So we are there where we think that there is no TB, whereas there is still no vaccine available. There are 14 drugs available for TB, but then there are mutations in bacteria when none of the drugs work. Mm-hmm. And the mortality rate is very high. It's much more scary than SARS-CoV-2. So I think the first start would be just to have poster like those where you make people aware. Like in 2020 alone, there were in 19, according to the recent WHO report, there were 10 million infections. According to WHO, there 1/3 of the world population has latent TB infection, and 10% of them will progress into active disease. So I think having that awareness is very important, because how can we attract any kind of fund until we actually prove that, actually show that there is a real problem, and what is the magnitude or gravity of that problem? I think that awareness is extremely important. And coming back to your second question, which is how can we avoid pandemic? I'm not sure we can. So, but we definitely have learned a lot. We all are so much more aware of sanitizing our hands. You imagine in January, we started from not touching your face and eyes to where every single child on this planet is actually talking about the term variant. So we have learned a lot from this pandemic. And I think I hope not, but then I think since the world is getting smaller and then everyone is traveling everywhere, we may have frequent pandemics, but then I think we just need to have a plan at place. And it is so exciting that what scientific community or world can do in a year. I mean, developing a vaccine which takes almost a decade, and imagining that so many people are already vaccinating, I think that is so reassuring that we got this. Yeah.
So how do you think we can push forward vaccinations for those infectious diseases that have been put on the back burner, like TB?
Funding is a huge challenge. Funding, I'm sure there will be people, there is this huge community who really want to and interested in working. TB research, the bacteria itself takes months to grow, months to culture. We need at least a biosafety level 3 facility, which is extremely expensive, and there are only few labs across the country where you can do these research. So I think funding would be one thing, and having that crosstalk between physicians and researchers and scientists from engineering, it's very important, because there are several aspects when it comes to vaccine designing or even tackling TB or another infectious diseases. And one of the basic thing is lack of point of care diagnostics. I mean, how do you perform a PCR test or any kind of test where you do not have electricity? How do you reach to all those places? If a test is costing you even $10 after subsidizing, how do you expect that a person a daily wages worker would actually go and get himself tested. So there is a lot that TB is another huge chapter, but I think having funding and having more awareness in people and realizing that TB is a global threat would be. The bacteria was discovered 5,000 years ago. It has been biggest killer in the history of mankind. It was isolated from mummies, and it has been there, and it has been mutating and growing stronger. So once we realize that, I think that would be a huge help.
Okay. Well, I think that is all the questions that I have from the audience at this point. Thank you so much for doing this. This was an excellent talk, and your answers have been very, very insightful, not just for me, but for everybody on the server. So thank you so much. Is there anything else you'd like to say to our attendees?
It has been such a pleasure to be here, and I wish you all good luck. And then you have my email address if you want to reach out anytime if you have any questions. I would be happy to help. And
thank you.
Yeah, have a good weekend. Thank you.
Thank you so much.
Yeah, take care.